Hi everyone, welcome back to BiochemVerse by Hammad. Today's lecture will focus on one of the most essential topics in biochemistry, water and pH. How biochemical buffer systems keep our body's chemistry stable. Everything that happens inside a cell, from enzyme reactions to energy transfer, occurs in an aqueous environment. So, understanding water and pH isn't just chemistry, it's the language of life itself. So let's move towards the lecture. So the importance of water in biochemistry, the significance of water in life. So think about it: every cell, every tissue, every organ in your body depends on water. Blood plasma is over 90% water. It carries oxygen, nutrients, and hormones to every level. Inside your cell, water acts not only as a medium but also as a reactant, like the medium for molecular transport and communication, and also reactant like and a hydrolyzing molecule during digestion. Even proteins and DNA fold into their correct shapes only in the presence of water molecules that stable their structure. So that was the basic of the significance of water in our life. And let's move towards the molecular structure of water. That includes the structure of water gives rise to all its special properties. Each hydrogen shares electrons with oxygen, but oxygen pulls them closer, making oxygen slightly negative and hydrogen slightly positive. So this polarity turns each molecule into a tiny magnet, and allowing water molecules to align, attract, and interact, forming hydrogen bonds that define the behavior of water in living systems. Enables hydrogen bonding between water molecules, that the polar molecule partial negative on oxygen and partial positive on oxygen hydrogen, sorry, and that's mix up. And so partial negative on oxygen and partial positive on hydrogen ends, and enables hydrogen bonding between water molecules that gives him many more properties of water, give rise to many other properties of water. That's basically because of the hydrogen bonding. So next is about hydrogen bonding and its biological significance, the secret powers of water. We can also say it like that: the uh, secret powers of water. That we just say that it's water, it's simple water. We are drinking water. Which powers does it have? And so there are also secret powers of water. That because of the hydrogen bonding. As previously I have mentioned that because of hydrogen bonding, it gives water more many abilities and many more secret powers. And so hydrogen bonds are weak individually, about 5% of the strength of a covalent bond, but collectively they are powerful. They make water cohesive. That's why water droplets form beads on leaves, and they give water high surface tension. That's how insects can walk on water. Lots of insects, because of they have a little weight. They are low at their weight, so because of the surface tension of water, and insects, small insects can walk on the water. So and on the molecular level, hydrogen bonds stabilize DNA, double helix, and protein folding, literally holding life together. If they are holding the protein and DNA and enzymes, then they are holding our life. That's also a point. So moving forward, the unique properties of water. Unique properties, as I have mentioned before, the unique properties that include high specific heat and high heat of vaporization, excellent solvent density anomaly. So let's discuss these. These properties make water life friendly. Life friendly. Water life friendly, not it's like water, then oppose, then life friendly. Water life friendly. So, because of its high specific heat, oceans can absorb huge amounts of solar energy without drastic temperature shifts. Because of high specific heat maintains body temperature, it also maintains our body temperature and also maintain the temperature of our environment of our earth. Like oceans can absorb huge amounts of solar energy without drastic temperature shifts, keeping global climate stable. So when we sweat, evaporation of water from skin takes away heat. That will help in maintaining our body temperature and perfect cooling mechanism. And unlike most liquids, solid water is less dense. That's why ice floats, forming an insulating layer that protects aquatic life in water. And let's move forward. The ionization of water. That's about the equation. It's all about the equation. The water undergoes autoionization. That water self-ionizes through slightly and through slightly. It's crucial. One in every 10 million water molecules splits into a hydrogen ion and a hydroxide ion. This equilibrium defines the concept of pH. We are moving towards the pH as our today's lecture was about water and pH. So this property, this equilibrium, that one in every 10 million water molecules split into a hydrogen ion and a hydroxide ion. This equilibrium defines the concept of pH, which expresses the hydrogen ion concentration of a solution. That the pH is basically what's the concentration of hydrogen ions in a solution, which expresses the hydrogen ion concentration of a solution, is known as pH, basis of the pH scale. Like we are moving forward toward the pH concept and we'll discuss it. The pH, as I have mentioned before, that pH is basically the expression of the hydrogen ion concentration of a specific solution, and is the pH. So the pH scale is from 0 to 14. First of all, you uh, take this concept that pH scale is from 0 to 14, and every step represents a tenfold change in hydrogen ion concentration. Tenfold means 10 raised to power 10 raised to power minus 14, 10 raised to power minus, and that's the 10 folds of each pH unit. As I have mentioned here, that each pH unit is equal to 10 cross change in hydrogen ion concentration. So each step represents a 10-fold change in hydrogen ion concentration. For instance, pH 3 is 10 times more acidic than pH 4. Like it's a scale 0 to 14, and below 7 it's acidic. As we come below the 7, 7, 6, 5, 4, 3, it's becoming more acidic. Like if the pH concentration, pH of the solution is 7, it's acidic. It's neutral. It's neutral, basically it's neutral. But as we move towards 0, like 6, 5, 6 is acidic, 5 is more acidic, and 4 is more than 5 acidic, and then 3, as I have mentioned, the pH 3 is 10 times more acidic than pH 4. So biological systems are extremely sensitive to pH. Even a change of 0.02 in blood pH can be fatal. pH and biological systems, like major biological buffer systems, we will discuss here. That the bicarbonate buffer system is crucial for blood, and when CO2 dissolves in water, it forms a carbonic acid, that and which can dissociate into hydrogen ion and HCO3 ion. And during exercise, when CO2 levels rise, this system shifts to keep pH constant. Inside cells, phosphate buffer handle metabolic acids and finally proteins, especially hemoglobin, act as buffer because amino acid chains can gain or lose protons. So enzyme activity is pH dependent. Extreme pH alter protein folding, denaturation, and cellular pH affects metabolism and signaling. So moving forward, acid bases and conjugate buffers. And so in biochemistry, basically acids and bases form conjugate pairs. One donates, one accepts. This dual behavior is what enables buffer system to resist pH change. Like acid and bases making a conjugate. Sorry for that. The previous pause was also because of this one. So acid and base pairs maintain equilibrium. Conjugate acid base pairs maintain equilibrium, like one is donating and one is accepting. Then they are maintaining the pH level. If we add more and more, then they will accept and donate. There will be cycle, equilibrium cycle, like carbonates, and then they are releasing hydrogen ion, and then they are accepting hydrogen ions, and this is maintaining the uh, equilibrium state. Like we are using the HCl and ammonia as a acid and base conjugate. So HCl is releasing hydrogen ions and ammonia is accepting that ion, so they are maintaining the pH, and they are not allowing the concentration of hydrogen ions to rise at such extent that they will increase even zero point 0.1 of the pH concentration of hydrogen ions. So moving forwards, the buffer systems. Buffer act like biochemical shock absorber, like they are absorbing the shocks of the biochemical systems. Like they stabilize pH when acid or bases are added. They are the key. There is a key lies in weak acid base pair that adjust ion concentration dynamically. As I have mentioned before, the we have discussed the HCl and ammonia example, that they how they are, one is donating and one is accepting the hydrogen ion. Then there will be when there is no free hydrogen ion, then there will be no rise in the pH. So the Henderson and Hasenbulge equation, where this equation tells us how effective a buffer is. When pH equals pKa value, the buffer can resist both acid and base addition equally. This is where biological buffer operate best. So that's the Henderson-Hasenbulge equation, and that relates pH to acid dissociation, predicts buffer behavior quantitatively, and at pH 1 equals to pKa, then there will be the buffer can resist both acid and base addition equally. So moving forward, the biological buffer systems. The human body uses multiple buffers. The bicarbonate system regulates blood pH. The phosphate buffers work inside cells, and protein like hemoglobin also absorb excess of the hydrogen ions to maintain this and the pH of our body. So these are the major biological buffer, the bicarbonate buffer systems, which are crucial for our blood. And as I have discussed this one before, that by absorbing CO2 in water, it forms a carbonic acid which can dissociate into hydrogen ion and HCO3 ion. And during exercise, when CO2 level rises, this system shifts to keep pH constant. So this one is related to the maintaining of pH in our blood. So the next is the phosphate buffer, which handles metabolic acids, regulates intercellular pH, and finally the proteins can also act as a buffer, especially hemoglobin, act as buffer because amino acid chains, side chains can gain or lose protein. The amino acid side chains. So amino acid act as a weak acid bases. So the next one is the bicarbonate buffer in action. How the bicarbonate buffer system works, because it's the main buffer in our body, because blood circulates in our whole body, and it will be a main buffer in our body, and it's controlled by the bicarbonate buffer. So bicarbonate buffer, this reaction, as mentioned before, the carbon dioxide and water, and they are making the bicarbonate, and then it's releasing hydrogen ion and maintaining the hydrogen ion concentration, and also simultaneously maintaining the pH of blood. So this reaction keeps your blood pH within a narrow window, and when you hold your breath, CO2 accumulates, pushing the reaction forward and lowering pH, making blood acidic. When you exhale deeply, CO2 leaves, pulling the reaction backward and raising pH. This delicate balance is an example of chemical homeostasis maintained by buffer. So the respiratory control and kidney regulation helps in the regulation of blood pH. So moving forward, the clinical relevance of buffers. In clinical biochemistry, even a zero point 0.1 drop in blood pH can be fatal. That's why buffer systems are critical to survival, and are constantly monitored in patients with metabolic disorders. And like if a patient has a metabolic disorder in pH, then they are regularly checked and maintained and monitored. So they are, if there are any change in pH, or acidosis, alkalosis, blood pH will be lower than 7.35, and alkalosis will be greater than 7.45, which with hyperventilations, diabetic ketoacidosis, and buffer failure, enzyme dysfunction, and organ failure, which will lead toward the buffer failure. Blood is not also working, and also other phosphate buffer, protein buffers are not working well. That's all because of the enzymes dysfunction and organ failure. If your liver is damaged, then your blood machinery is shut down. Not properly shut down, but they will. That will be an instance. There will be example that organ failure can do many things. Like, the summary, to summarize, water provides the medium of life, and pH defines the balance, and buffers guard it. Without this trio, no biological process could function. As so, to sum up, water isn't just a liquid, it's life most critical medium. Its polarity and hydrogen bonding make it the perfect solvent, and its buffer systems ensure our biochemistry stays balanced. For maintaining blood pH to stabilizing enzyme, water is the silent guardian of life. So that was the summary, and that was the lecture. So in the next lecture, we will discuss about the carbohydrate. We're moving towards the basics of the biochemistry, and biomolecules, carbohydrates, proteins, nucleic acids, and lipids. Then we'll move towards their and that's the on the slides. I will also manage to do it on the paper or on the board and record the videos when we will discuss about the metabolism, that because that's a tricky one, and we will do our best to educate you and relate everything. So the coming next is this one. So we'll move to biomolecules, starting with carbohydrates, the energy currency of life. So don't forget to like and comment your questions, and also subscribe for more lectures in upcoming videos. So like and subscribe our channel on YouTube and also on the Facebook. It's with the name Hammad Bashir Khan Baloch, and on the YouTube it's BiochemVerse by Hammad. So subscribe it, and also follow us on Instagram, BiochemVerse, and also the links in the description. And if you want the slides, then there is also a link in description. You will follow it and make your request for the presentation of the lecture, and we'll do our best for your assistance.